Dr. Baliga here. This podcast is on SolMER as a potential biomarker for cardiotoxicity. SolMER is soluble myeloid epithelial reproductive tyrosine kinase receptor. This is a receptor on the surface of cardiac macrophage and is a key to phosphatidylserine triggered activation of the cardiac macrophage for cardiac recovery and after injury. What prompted me to do this podcast is an exciting paper by Nicolás-Ávila et al. Titled "A Network of Macrophages Supports Mitochondrial Homeostasis in the Heart" in the journal Cell 2020, volume 183, pages 94 to 109. It's exciting because Nicolás-Ávila et al. Show that macrophages form an important support staff to cardiac myocytes. Cardiac myocytes contain mitochondria to contract continuously over the years, but as the mitochondria decay, they are ejected from the myocytes and they are mopped up by the macrophages resident in the myocardium, and they serve as a support staff, a kind of custodians which mop up after all the mitochondria are released from the myocytes. Macrophages in the embryo populate tissues during development and self-renew or are replenished regularly from the bone marrow. Myeloid progenitor cells from the bone marrow are released into the circulation. They then migrate to the spleen, where extramedullary monocytopoiesis takes place to replenish the pool of monocytes, which can then be rapidly mobilized to the damaged heart, and this is known as the cardiosplenic axis. Therefore, the macrophage lineages in the heart arise either from the yolk sac or the hematopoietic stem cells, both embryonic and adult. Nicolás-Ávila et al. In their Cell paper showed that each cardiac myocyte touches on an average 5 macrophages. Therefore, the authors evaluated the relevance of macrophages to cardiac function, and what did they find? Using sophisticated techniques and markers, they found that the mitochondria are actually ejected from the cardiac myocytes and are engulfed by this backbone of nearby cardiac macrophages. In addition, they showed that the packets of ejected mitochondria are encapsulated in structures called exophores. These exophores in the cardiac myocyte are highly enriched for mitochondrial proteins relative to other cardiac myocyte proteins such as sarcomeric motor proteins. However, just days after gulping the exophores full of cardiac myocyte mitochondria, the macrophages no longer expressed these mitochondrial proteins, suggesting that they fully digested these spent mitochondria. Clearly, the process of mitochondrial transfer from the cardiac myocyte to the macrophage requires a normally functioning process used by cells to digest organelles, the process of autophagy. When the cardiac myocytes are exposed to cardiotoxicity, there is an increase in exophore production. This reinforces the concept that cardiac macrophages are needed to clear spent mitochondria. The exophores are marked by phosphatidylserine, which serves as a signal to the macrophages to ingest the mitochondria. phosphatidylserine is often present on the internal face of the membranes, and when cell injury occurs, this moiety flips to the exterior surface of the membranes, where it can trigger activation of neighboring cells. Myeloid epithelial reproductive tyrosine kinase receptor, MERTK, is a receptor on the surface of the cardiac macrophage and is vital to phosphatidylserine triggered activation, and has been shown previously by Ed Thorp's group at Northwestern that it is required for cardiac recovery after injury. Nicolás-Ávila et al. In the Cell paper showed that in mice lacking MERTK, cardiac macrophages were intact but were unable to absorb mitochondria, a finding that implicates MERTK in the removal of mitochondria. The extracellular portion of MERTK can be cleaved and released in the serum, where it can be measured as a biomarker in patients after myocardial infarction and possibly after chemotherapy-induced cardiac damage. Traditionally, we have viewed macrophages as carriers of interferons causing myocarditis, but this new exciting paper suggests that macrophages serve up as a support staff, as a quality control mechanism by which these cells contribute to metabolism with exciting biological implications and even potential therapeutic value for various heart diseases. Another potential biomarker is imaging stem cells and these macrophages, exciting work done by Dr. Jagat Narula's group at Mount Sinai. So together with soluble myeloid epithelial reproductive tyrosine kinase receptor may be the way forward in diagnosing and treating cardiotoxic side effects. In summary, Nicolás-Ávila et al. Have discovered a novel mechanism by which cardiac myocytes sustain mitochondrial quality and cardiac function. They found that cardiac myocytes rely on a network of resident cardiac macrophages for clearing dysfunctional cellular parts through so-called exophores, which is an ejection mechanism for large vesicular particles. This seems to occur in cells which might not have a capacity for common self-renewal and regenerative pathways, and therefore rely on these waste disposal mechanisms. Other cell types which use exophore shedding include skeletal myocytes, brown adipocytes, and neurons, all of which display high mitochondrial content and are highly metabolically active. So the biomarkers are imaging of stem cell in the heart, including macrophages, and soluble myeloid epithelial reproductive tyrosine kinase receptor.